there's also space, and then there's time, which is some of the molecules are really long-lived, some are really short-lived, right? And so we believe that the bacteria have access to all of that information, right? We just know they're machines at extracting both the amounts of those molecules, but the physical properties, which is what you're getting at, and rates, right? So if you make it faster, right, or slower, that means something to you. And so all of these feedback loops, exactly like in an engineering, computer circuit, exist. Like if I perceive my molecule, they put more receptors out there so they can pay better attention to it, right? We know those things are encoded in these circuits, right, that gives them, it's a much more complicated computation than just the molecules there, the molecules not, right? But we don't know the full answer to that because we're just now starting to think about this temporal and spatial, and we can do things like if we knock out these feedback loops so that you can't amplify the signal, we see that they get kind of asynchronous in their behavior, right? So that's what we do. But I think that is the level that we're getting to with these, besides always hunting for new molecules. We are doing that. But I think that part of it, like I think even if we never find another molecule, which we already have one, so anyway, if we never find another one, I think even with just these two, trying to figure out how they maximize information input, right? We're quite busy because of all the tricks that they've put into these circuits, like reducing noise, like, things like that are in there, and just with two keeps us busy, right? But it's very fun to think about that level. And we could never do that before because we didn't even have any of the molecules. But now I think we're getting a little more sophisticated in our thinking about, like we look at those two molecules and we're like, even though they're all small molecules, these molecules, the two I showed you, the intra and the interspecies, are so different physically. So you think, well, there's a reason those evolved to have these different, one goes really far and one, so I think that's very interesting to think about.
So I keep thinking about this little exhibit that we have here on the evolution of mutants. And have you seen, since you've extracted mutants, and how do they you have to keep a certain number of mutations within your population. And so is there also a communication there?
Yeah.
And if we develop a drug that blocks them, the bacteria are going to respond to that.
Right.
And then we might be in the same problem that we're having now with antibiotics.
But I already told you that I need a job, right? I'm only halfway done, right? So that's a fabulous question too. All right, so let's see, there was multiple questions here. So the first one is, right, so you guys know mutations just happen spontaneously. So you have 10 to the 9th bacterial cells, you have mutants in there, right? And so occasionally, so forget about the drug for now, just quorum sensing. You're thinking about how quorum sensing got on this earth, right? You have mutants, right? Like the best thing, like I always think if I were a bacterium, what I would do is I would make the signal and I would make you turn on the 100 genes, right? Because that's what costs, not the signal. The molecule's cheap. Turning on these 100 behaviors where you're giving this stuff away, that's expensive, right? And so mutants arise, absolutely. But if so many mutants arise that quorum sensing, doesn't work, right? They're not working as a collective, right? There wouldn't be quorum sensing, right? And so what we don't know is the answer to your question, of course, is how do you keep cheaters? There have to be molecular police, that there's some punishment, if you will, meaning you don't get to grow if you're a cheater, right? And I can tell you examples. For example, in that light organ of the squid, you never find a dark mutant. So the squid has a mechanism for being, just like this immune system business, it can tell the bright ones from the dark ones, and the dark ones go, right? And so there have to be somehow built into these systems management, I'll say, of cheaters, because they are arising all the time. And then when for quorum sensing to be here, the benefits have to outweigh those costs, right? And so we know quorum sensing's here. So somehow, right, evolution has a way of controlling cheaters. And so now the evolutionary biologists are into this because they want to figure that out, right? And so that's very, they used to like work on, a herd of wildebeest, right? But now they can really actually make mutants and do these tests. So they're really into this micro stuff now. And then the second question is, let's say we can make this fantasy anti-quorum sensing drug, right? You're going to get mutants that are resistant. So anybody who changes its receptor so it doesn't no longer is susceptible to the drug. So we know resistance will evolve. They're bacteria, resistance will evolve, right? And so all we're trying to do with this drug is to buy time until someone figures out the next behavior, like maybe biofilms or something to conquer. It's not, this is forever, that we have to keep making these things. What is fun about the quorum sensing aspect of this is we don't know how they're going to get resistant, right? Like, so if you have a traditional antibiotic, and you can replicate your DNA in the, you're resistant, right? But here, if you become impervious to the drug, you still have to be able to detect the real signal. So that's a hard mutation to get, where the drug doesn't affect, but the real signal works fine. And then also, let's just say you're in this population that's being treated with this drug, and most of the guys in the population are susceptible, but you're not. And so what do? You turn on, it's what you said, you turn on your quorum sensing regulon, but nobody else is participating, right? And so you actually are at a selective disadvantage because you're working, but you're not getting the benefit of the collective. So they will get resistant, you're right. It's not going to be by the obvious way of simply getting around the drug. It has to be something else, and we don't know what that is yet. But what's really fun is now that we have the drugs, what you can just do is put them on bacteria on Petri plate and ask for a guy that turns on light and see what's wrong with it, right? So that we had to have those molecules to even ask what's the mechanism of resistance, the possible mechanisms of resistance. So we're actually trying to make them, sounds so stupid, we make the drug and we make them resistant in the lab so that we can figure out what's going to come down when we actually, right? So we're trying to do that now. Have I exhausted you? Yes. Uh-oh, no, I have not. Good.
So my question kind of goes back to the length and shape of the actual molecules. So it kind of makes sense that the interspecies communication molecules are hydrophilic and travel. But is there any kind of correlation between the intraspecies molecules that, like if they have a longer tail, does that mean that they're more pathogenic or?
Right, that's a good question. So I don't know if there's a pattern like that. So for sure, you're right. The longer the tail, the more hydrophobic, so the less it travels. Right? So that's the greasier it is, right? And that's a good question, and I don't know if anybody's ever actually tried to map that out to say, you cause this disease and you make a molecule that looks like X, or but that's an interesting idea, and I don't know if anybody's done that. Yeah, it's a great idea, right? And we have, we have a few more molecules than what I've shown you, and so you could actually make such a map. You could do that. I don't think it's been done. Yeah, but that's a good idea. I'll get on my cell phone. Yeah. Yeah. Thank you, you guys. You're a really great audience. Thanks for coming.